Okay, so the system that I'm going to be talking about is a system that we've worked on for some time in my lab, which is in smooth muscle cells. And in particular, the differences in phenotype between the sort of contractile form of smooth muscle cells, which you find in blood vessel walls, so vascular smooth muscle, but also in visceral smooth muscle. So the transition between a contractile phenotype and the so-called synthetic phenotype. So these are marked by, obviously, the contractile phenotype. It is contractile. It doesn't show much migratory activity, and it's low proliferation. Whereas this synthetic phenotype is more proliferative, migratory, non-contractile, and it's called synthetic because you get a lot of synthesis of extracellular matrix components. And this transition is of biomedical interest because, for instance, it occurs in atherosclerotic plaques. You tend to find this synthetic phenotype there. Now, a lot's known about the control of the gene expression program at the level of transcription, the role of myocardium and serum response factor. The role of microRNAs is also known. But obviously, we've been interested in the role of alternative splicing during this transition. Now, up till now, we've known only about a couple of model systems, and in particular, in my lab and one or two other labs, a small number of model systems have been looked at. These are two of the ones that we've looked at in my lab, and while we know quite a lot about the details of what happens in these particular exons, it's not clear always what the general principles of the regulated splicing program, what those principles are. So for instance, there's a single example here. In the alpha-tropomyosin gene, PTB, polypyrimidine trap binding protein, is particularly active in the contractile state, where it represses the exon that's usually used in as a default in other types of cell. Whereas in the actinin gene, the same protein, PTB, is active outside of contractile cells, and it keeps this smooth muscle-specific exon repressed, except for in the contractile phenotype. So which is the more typical of this splicing program? So to try to address this, we've taken a more global approach using the Affymetrix exon or junction arrays, the MJ array, which first became available to us via the EURASNET. What we've done is, because these are only available for mouse and human, we chose to use mouse, although rat is actually a better established model system for vascular smooth muscle. And what we've done is to compare the smooth muscle cells that are actually present in tissue. So we can strip the tissue so that we've only got a smooth muscle layer in the media of the aorta. And we can compare the RNA isolated from these intact tissue here, so still embedded in extracellular matrix. And we can compare that with cells that have been put into culture, and in this case, it's just for 7 days. So this would be more the synthetic phenotype. And we actually did this for both aorta and bladder. The reason we did this is that physiologically, they're rather different types of smooth muscle. Aorta is a more tonic form of smooth muscle, which has constant contraction, maintains the tone of the vessels, whereas bladder is a type of smooth muscle which doesn't contract for most of the time, but then in short bursts it will contract. So they're rather different. But we previously knew that the alternative splicing events that I showed before are both highly regulated in those two tissues. So we then compared the contractile cells with the synthetic cells. These were RNAs hybridized to the MJ arrays. The data was analyzed by Melis Kayeki in Yone Ule's lab. And we were then able to look at the transcript level changes, so changes in gene expression levels, and also splicing changes. And in both tissues, we could find large numbers of splicing events that were changed. By and large, I'm only going to be talking about the data from aorta, but generally we've got similar pictures in bladder. So nearly 3,000 upregulated events in contractile cells and about 1,500 in the synthetic cells. So these are just two examples of them. This is actually the alpha-actinin gene that I'd already told you about. So the array rediscovered this event. This is a mutually exclusive exon pair. In both the aorta and the bladder contractile cells, you only get that SM exon included. But in the synthetic cells, you see a substantial switch to use of the alternative exon. So that was one we already knew about. Here's another example. This is an exon in the smoothilin gene, which is actually mainly skipped in the contractile cells. And then you get a complete switch to inclusion of the exon in the synthetic cells. And we've validated a large number of other examples. But these are just two examples of the kind of splicing changes that we see. And as you see, it's a very similar picture in the two different tissues. So we then looked at the the functions, the ways in which transcription changes and splicing were remodeling the transcriptome. And we found something which Ben Blanco and others have described a number of times, which is that changes at the level of transcription and changes at the level of alternative splicing remodel different parts of the transcriptome. So for instance, the genes that were affected by alternative splicing were very highly enriched for cytoskeletal proteins, and in particular actin family cytoskeletal proteins were being heavily remodeled by alternative splicing. In contrast, at the transcriptional level, transcription that's upregulated in the synthetic cells involves signaling molecules, extracellular matrix proteins, as we would expect, and growth factors. Whereas in the contractile cells, the main upregulated class of genes were voltage-gated ion channels, as you'd expect from these contractile cells. But also, interestingly, there was a very, very significant depletion in the transcriptionally regulated genes of nucleic acid binding proteins. And in particular, RNA binding proteins are very heavily underrepresented amongst the genes that are altered by transcription. These actually also did feature over here. They were also depleted in the genes that were upregulated in the other direction, although the significance wasn't as large as here. Okay, so this was the general roles of the kind of genes that splicing was affecting. But we also then looked at the subset of events that were affected by intron retention. So what we found here are a couple of things. The first is that if we looked at the intron retention events, we found that there was a very asymmetric distribution. So there was a much larger number of introns that were more retained in the contractile type cells, and a much smaller number that showed higher retention in the synthetic type of cells. Now, when we looked at the functional enrichment of these genes, we first of all found that actually the genes, the annotated intron retention events that were present on the array were already enriched for RNA binding proteins. But if we used all of those events as a background set and then looked at the functional enrichment here, we found that mRNA processing factors were highly enriched amongst these sets of intron retention events. So a number of names that you'll recognize there, various SR proteins, U2 SNRP proteins, but not just splicing proteins. So for instance, this is the adenosine methyltransferase that can modify A residues, and we've got 3' M processing factors there as well. So there's more intron retention, which generally is predicted to downregulate expression of these proteins in the contractile cells. Just as one example, I said SRSF1 there is at the top. Actually, this was one where there were two separate events, one of which was much more retained in the contractile cells and one which was more retained in the synthetic cells. But as I'll show in a minute, although the intron retention is going in the opposite direction, the effect upon expression is actually collaborating and pushing in the same direction. Now, this is hinting already that maybe the general splicing activity is lower in the contractile cells. But if we look at the distribution of cassette exons, and I should have said that these plots here are simply using the DI rank statistic, which gives an idea of the confidence of a change in direction of splicing between the cells. So the further to the right, the more confident we are that those events are shifting in that direction. If we look at, although intron retention was much more prevalent in the contractile cells, when we look at cassette exons, they're more balanced. So although there may be possibly a weaker splicing environment in contractile cells, this group of cassette exons seem to be perfectly happy. In fact, there are more cassettes that are included in contractile than are upregulated in the synthetic cells. Now, in one of the projects in the lab, we're particularly interested in using this set of exons to look at, what are the general properties of smooth muscle regulated exons. And Nicolas Bellara in Eduardo Iris's lab has been looking at motif enrichments in particular around these kind of exons, and it actually suggests that repression of these exons by PTB outside of the contractile cells is the more general feature. But for today I want to stick to this idea of the non-productive splicing. So when we went back and looked at all of the splicing events, apart from intron retention, we found that many different types of non-productive alternative splicing event were affecting splicing activators, SR proteins, but also core U2 SNRP components, some U1 SNRP components, U170K, and also the regulatory kinases. So what I'm going to do now is just go through some of these events and show that the array predictions were correct. But the key thing is, in every case, the alternative splicing event is predicted to downregulate expression in the contractile phenotype. So first example, SNRPA1. So this is the U2A prime protein. There's a pair of retained introns here. In both cases, the intron retention is higher in the contractile cells, and the productively spliced version is higher in the synthetic cells. Now, I've shown here one Western. I'll mention a little bit later. We had problems validating the protein level changes simply because of the nature of the biological material and getting enough of it. This was one case where with the bladder tissue, we were able to see much higher levels of the protein in the synthetic bladder cells compared with the contractile cells. So this is a pair of intron retention events. Actually, in a cell line, we've also seen an exon skipping event, which is also non-functional, which is more prevalent in the contractile cells. This is the SRSF1 set of events. So in this case, we've got two intron retention events. Intron 3 is more retained in the contractile cells, and work in Adrian Krainer's lab previously showed that retention of intron 3, although it prematurely terminates the open reading frame, and if it were translated, you get a protein with the RRMs, but without the RS domain, so that would have altered function. But Adrian's lab showed that actually when you retain intron 3, that RNA is nuclear retained, so it's down-regulating it. There's then intron 4 is regulated in the other direction. That's more retained in the contractile cells. Sorry, no, it's more retained in the synthetic cells, more spliced in the contractile cells. But this is an event where when it's spliced, it would provoke NMD. So the fact that you're getting a higher retention now in the synthetic cells would help to upregulate it. So although the intron retention events are going in the opposite direction to each other, they're combining to give higher expression in the more proliferative synthetic cells. A couple of examples of poison cassette exons. There's a well-characterized poison cassette exon in TRA2 beta that Stefan Stamm worked on a number of years ago. Again, this poison exon is more included in the contractile than the synthetic cells. Likewise, a poison cassette exon in SF3B3 is more included in the contractile than the synthetic cells. Last example I think I'm going to show is an event that actually was very nicely described in a very recent paper by Albrecht Bindereif, where they looked at an autoregulatory circuit that couples U1C and U170K expression. So again, in this case, we've got two events, either inclusion of a poison cassette exon, which Albrecht showed leads to NMD, or indeed use of an internal poly-A site. And we find that both of those non-productive events are at higher levels in contractile cells, and the productive splice is significantly enhanced in the more synthetic cells. And we've got at least a fourfold increased expression there. So we've got a number of these non-productive alternative splicing events. If we look at the transcript levels, we actually don't, with one or two exceptions, we don't see dramatic changes in the levels of the transcripts there, which we don't fully understand, given that some of the splicing events would be predicted to lead to NMD, but it could be that the activity of NMD is variable between the two cell states. Now, obviously, what we really want to do is to look at the, no, let me just back up a minute. So in this is a victim of last-minute resorting of slides this morning. So we've got these non-productive splicing events that predict lower levels of the protein in the contractile cells, but we had a big surprise when we looked at the levels of the snRNAs in the same cells, because we saw higher levels of the snRNAs in the contractile cells, where you had the non-productive splicing, and significantly decreased levels of those snRNAs in the more proliferative cells. So there's a disconnect here between the levels of the splicing factors, which included SNRP proteins, U170K, U2A prime, SF3B proteins. There's a disconnect between those and the levels of their cognate snRNAs. Now, the missing link we have at the moment is the level of the proteins. As I said, doing quantitative Westerns, or any Westerns actually, on mouse aortas, intact aortas, was really challenging. So what we've done is shifted to using another model system, which is rats, which are a lot easier to work with and have been much more worked on by the cardiovascular biologists. And we can actually get 4 different types of sample here. We can have our intact tissue. We can also enzymatically disperse the cells and harvest enough single cells at day 0. We can then also put them into culture and take very early passage 0 cells, or we can let them passage for a number of times. Now, actually the transition between passage 0 and passage 9 has been extensively used by cardiovascular biologists to look at phenotypic modulation. So what we're aiming to do is to actually collect RNA and protein samples from all 4 of these states, do RNA-seq to get the transcriptome data, but then also do quantitative proteomics by selective reaction monitoring so that we can get absolute quantitation on these various spliceosomal proteins and activators that we've looked at. Now, we don't have that data yet. That's work in progress. But what we do have, Dipen Rajgor in Kathy Shanahan's lab in collaboration with us, has done immunofluorescence microscopy comparing these 2 states, which in the rat are, this has been typically taken as a differentiated state, and this is a more synthetic state. And we've looked at expression of the various splicing factors. And what we see is quite dramatic changes in their expression. So I'm going to I'll go through this quite quickly because all of the images are rather similar. In each case, on the top row, we've got the more contractile cells, the passage 0. And on the lower row, we've got the passage 9 cells, which are more synthetic. And we've always got the immunofluorescence, then an emerge of the immunofluorescence and the DAPI, and that's just to prove there were cells on the top there. So what should be immediately clear here is we've got very dramatic differences in the levels of U170K protein, also in this case the U2A protein, very dramatic differences between the contractile and the synthetic cells. And the next few slides are just going to show this is the same for many other proteins, SF3B1, SF3B3. It almost, I mean, one's tempted to say it's an on-off switch. It almost certainly isn't because splicing is occurring in these cells, but it's a dramatic change in expression. SRSF6, which is SRP55, TRA2 beta, again, as we had expected, large changes. This is the change in SRSF1. Again, a large change there. Now, as a sanity check, we needed to show that some things weren't changing, and RNA polymerase II actually is present there at similar levels in both phenotypes of the cells, as indeed is SMD, one of the coarse NERT proteins that we've looked at here. So it really is, it's not, as I said, the polymerase is still there, and coarse NERT proteins are there at similar levels. We also found, to our surprise, that some of the auxiliary RNA binding proteins, which are known to regulate splicing, in this case PTB and muscleblind, which we know are involved in regulating, for instance, the alpha-tropomyosin event, those were there at similar levels in those two cellular phenotypes. So I think that's all the data. So the conclusions as they stand at the moment, we can see much higher levels of non-productive splicing of many core splicing factors and splicing activators in this contractile phenotype of smooth muscle cells. So there's a concerted program of all of those factors together, but it seems to be associated with lower protein levels of these core splicing factors and activators. And I think that's a key point to make, because many people have looked at these non-productive splicing patterns before, but they've always been thought of as sort of rheostats, as things to prevent overexpression. So you'd expect to see more non-productive splicing when the level of the protein is high. But in this case, we're seeing them going together, which suggests there's some other signaling mechanism that's feeding in and in a concerted way damping down the expression. So the intron retention was more prevalent in the contractile cells, but importantly, it's not just that splicing is not happening or is rubbish, because many cassette exons, actually the majority, are more included in those cells. And then finally here we've got the fact that there's higher expression of the snRNAs in the contractile cells, even though the cognate proteins are lower. So we think this suggests a number of things. I mean, in regulation of alternative splicing, many of us in the field have tended to think, well, you've got your functional spliceosome components, but what makes the difference between cell phenotypes is the changes in the levels of auxiliary factors. Now, a number of people have suggested over the years that it could be that knockdowns, for instance, of core splicing factors can change alternative splicing patterns. And indeed, we think in this particular cell phenotype or phenotype transition, changes in the core splicing machinery could be important in modulating a number of splicing patterns. And more than that, we think actually the balance between the snRNAs and their cognate proteins could also be important in determining the state of the SNRPs in those different cell types. So clearly there's a number of unanswered questions that we're intending to pursue from this, but for now I think I should leave it. So the acknowledgements, the people in my lab, this project was started by Martina Hallager, who actually left the lab about 2.5 years ago, but it's been continued by Adrian Buckroyd, Miriam, Claire, and Selena, and we've also had important collaborations with Kathy Shanahan's lab, Dipen Rajgor, who's actually going to be joining our lab in a couple of months. Yone and Melis have been very helpful, as has Nicola and Eduardo Iras. And funding has mainly come from the Wellcome Trust, and I will leave it there and let you get to coffee.